Polypeptides are amino acids that are connected to each other with amide functional groups, meaning that the amino group on one condenses with the carboxylic acid on the other to form an amide functional group, which is called a peptide bond. Polypeptides have amino acids linked one after another in long chains, hundreds sometimes, of amino acids, one right after another.
Here is an example of a polypeptide, and this polypeptide has in order free amino acid on arginine, and then connected to alanine, and then connected to glycine, and then connected to serine, and then connected to phenylalanine, and then to another phenylalanine, and then to leucine. And the leucine has on it a free carboxylic acid group. The arginine is attached to the alanine by the carboxylic acid group on the arginine to the amino group on the alanine. The alanine is attached to the glycine by the alanine carboxylic acid group attached to the glycine amino group, and on across the chain. You always list the amino acid with what we call the N-terminal amino acid group first, meaning that the amino group is free on that end. And we always write the C-terminal amino acid last. And C-terminal means there's a carboxylic acid group free on the other end. The order of the amino acids in the polypeptide is called the primary structure. Because a polypeptide is so large, there are a very large number of amino acids in polypeptides. Because it's so large, its three-dimensional shape is very complicated, and so we describe the three-dimensional shape in 4 ways. The first way we describe the three-dimensional shape of the polypeptide is by telling what is its amino acid sequence. So we call that the primary structure of a polypeptide. There is hydrogen bonding between amino acids in the chain that often will cause a regular repeating three-dimensional structure. An example of that is that some proteins will hydrogen bond themselves into an alpha helix structure. Some will hydrogen bond themselves into a what's called a beta-pleated sheet. In a beta-pleated sheet, you actually have, it's kind of like folded paper. There are chains of polypeptides that are hydrogen bound to each other lengthwise, and they actually get into, because of the rigid polypeptide structure, the rigid peptide bond, you actually get a very rigid structure that looks like an accordion-shaped piece of paper. It's often called a pleated sheet. The tertiary structure is the interaction between amino acids in a chain causing a three-dimensional globular shape. Oftentimes, where they're showing you a polypeptide, you will see that it looks like someone took a chain and wadded it up. That's the three-dimensional globular shape that they're talking about. Then there's quaternary structure, and quaternary structure is how one long chain of amino acids, one polypeptide, will often have intermolecular attractive forces, or sometimes even covalent bonds between them, and they will associate together and form a three-dimensional structure where they're hugging up close to each other. That is two chains forming an additional three-dimensional structure called the quaternary structure. So primary amino acids sequence, secondary hydrogen bonding causing a regular repeating three-dimensional structure, and that's either an alpha helix or a beta pleated sheet. Tertiary structure, which is how the chain will wrap around onto itself and cause a three-dimensional globular shape. And then quaternary structure, which is the interaction of two chains hugging up to each other. Can be more than two chains. Okay, here is a picture of secondary structure, and you can see how this is a regular repeating hydrogen bond between two chains, and this is actually what it looks like between two chains in a beta pleated sheet. In a beta pleated sheet, what you usually often will have is more than one chain, though. For instance, if you look at collagen, it's many chains running together to give you this flat, stiff structure in collagen. A polypeptide can fold and coil into that three-dimensional globular shape, and there's many ways that it can do this tertiary structure. We've already seen that two thiols can be oxidized to form a disulfide linkage. Cysteine, the amino acid cysteine, has as its R group a thiol. The thiol in cysteine can be oxidized and form a disulfide that forms a very strong link between one part of a chain and another. You can have hydrogen bonding between side groups and peptide bonds, or peptide bond to peptide bond. If it's not regular and repeating, well, then it's determining the tertiary structure. Also, an ammonium group on one side chain can be attracted to a carboxylic acid group on another chain because they're ions. The ammonium will have a positive charge and the carboxylate group will have a negative charge, and they would be attracted to each other because their charges are opposite, and they can help draw these chains together. And then there is hydrophobic to hydrophobic conjugation. Something like leucine can be attracted to another amino acid chain. The side chain on leucine can be attracted to another side chain that is hydrophobic, like leucine. An example would be isoleucine or phenylalanine. They have hydrophobic side chains, and they will try to group together so as to maximize intermolecular attractiveness. Here is a picture of how a disulfide linkage can occur between two cysteines. I'm drawing the side chains of cysteines, and you can see the thiol group. There's one cysteine on the left and another cysteine on the right that are in a polypeptide chain, and there is a thiol group on either. If they are oxidized, they form this disulfide linkage, which is a strong connection between one part of the amino acid and another. Enzymes are the catalysts in our body that control our biochemical reactions. Most enzymes are polypeptides. Enzymes are catalysts, so they participate in the reaction and they lower the amount of energy that is needed so the reaction will speed up. But they are not consumed by the reaction, they only participate in them. The way that enzymes are constructed is they are these polypeptides that have this three-dimensional shape, often globular, so that they have a little site inside them formed by their structure. That site inside this globular nature allows a substrate to come in, or maybe sometimes two substrates, to come in, sit down, and then be modified by the enzyme. Many enzymes have a cofactor that is needed for activity, that they're inactive unless they have this cofactor associated with them. There are some that are organic molecules and some are inorganic ions. If the reaction is an endothermic reaction, The enzyme will use ATP to do the reaction to change the molecule, and it'll take ADP and cleave a bond off it and make ADP and take the energy that was in that phosphate bond to conduct the reaction. Sometimes they'll take several ATP to do a particular reaction. The enzymes' active sites are formed by their three-dimensional shape, and there is one way of describing this as a lock and key model of how the enzyme works, that in the same way that a key will slide into a lock and fit perfectly and be able to turn the lock, substrates can slide into a portion of the three-dimensional shape of the enzyme, fit perfectly, and then the enzyme can modify their shape. There are some other models of how enzymes work. There are inhibitors that can compete with your substrate coming in, and the inhibitors can either sit down in the site and block the site, or they can fit somewhere else on the enzyme and cause the enzyme to change the shape of its active site. And so you do have inhibition of enzymes, which is good because that's how enzymes are controlled in making reactions go. So the inhibitors slow down the reaction, and so when certain things build up, your enzyme might be inhibited. When certain things are in low concentration, then it would be put back into action. Denaturation is the loss of the three-dimensional shape of a globular protein. That three-dimensional shape is essential for its activity because it forms its active site. When a globular protein loses its three-dimensional shape, it is no longer active. This three-dimensional shape, the tertiary structure, is dependent on many bonds. Now, the disulfide linkage is hard to break, but hydrogen bonds are very easy to break. Ionic bonds between carboxylic acids and amine groups are very easy to break, and the hydrophobic association between hydrophobic groups is very easy to break. The three-dimensional shape of an enzyme can be lost by having it go into certain conditions. Denaturation can occur relatively easily. The tertiary structure of a protein can be easily changed by just changes in pH or changes in temperature. There's usually a narrow range in which a narrow range of pH and temperature in which the tertiary structure is stable. Solvent reactions can also do this. The reactions, especially redox reactions, will change the functional groups in the amino acids. Those functional groups are important for the tertiary structure. So redox reactions, or any kind of reactions, but particularly redox reactions, will change that tertiary structure. Particularly susceptible to redox reactions are the disulfide linkages, because if we reduce a disulfide linkage, the disulfide linkage comes apart and forms the two thiols. Oxidation of an enzyme can actually form a disulfide linkage where you don't want one. There are small changes only necessary for an enzyme to lose its active site shape, and then it's no longer active. So tertiary structure is incredibly tenuous, meaning easily disturbed, and is very important to the activity of the enzyme. Whenever change in tertiary structure occurs, it is almost always irreversible. An example of seeing denaturation occur is when you cook an egg. Eggs have a large amount of protein in them, and when you cook it, you can actually see the proteins changing. because the consistency of the egg and its your ability to see through the egg changes as it cooks. Cooking is important for denaturation because if you have microbes in food, when you cook the food, you heat it up and denature the proteins in those microorganisms, and then they die. Be sure to do in-text problems as you read, and do the following end of chapter problems. I will be posting an online quiz for you to complete.